Hi, everybody. Today I'm going to talk a little bit about genetics, sort of setting up some of the basic principles that you'll be hearing about as we go through a lot of these chapters. I have to say genetics is probably not one of the major topics covered in biopsychology, but it does sort of underlie a lot of what we talk about. We talk a lot about the brain in biopsychology, and the brain is formed based on the instructions in our genes. So are our hormones, so is our sensory organs and our muscles. Everything about us comes from genes. So sometimes it feels like it gets a little bit lost in some biopsychology, but just remember, genetics is underlying all of these biological processes that we're going to talk about throughout the next several weeks. So let's get started. So starting out, we're just going to go over some basic definitions of like the structures and the molecules that you really need to know and sort of how they all tie together. So starting literally at the beginning, cells. Cells are in all living things. They're the things that promote growth, sustain life, and everything that happens in cells happens due to basically an instruction manual in the form of DNA. DNA really can be thought of as an instruction manual for how to make everything your body needs, whether it be a cell membrane or a receptor on a cell or a muscle fiber or a neuron. DNA tells your body how to do all of that. So chromosomes are found within the nucleus of the cell. And chromosomes are basically tightly woven DNA, so DNA that's been compacted together. And all humans, well, most humans have 46 chromosomes arranged in 23 pairs. So if you've ever heard of the service 23andMe, the 23 is for these 23 pairs of chromosomes. 22 pairs are what's known as autosomes, or the things that sort of have all the instructions common to all humans, but then the 23rd pair is actually your sex chromosomes, and that is what either makes you male or female. And the chromosomes are made up of DNA, which stands for deoxyribonucleic acid. So this is a molecule, like I said, that includes all the instructions that you need to manufacture everything your body needs. These are written, technically they're written in a 4-letter language, so that it's composed of 4 chemicals. So that's why it makes sense to think of it as an instruction manual written in a 4-letter language. These are usually just abbreviated A, C, T, and G, or adenine, guanine, thymine, and cytosine. So this is what a picture of what this would look like. So on your left, you have the cell. The dark purple on the inside is the nucleus. So if you zoom in on the nucleus, you would see, not lined up neatly like this, but you would see 23 pairs of chromosomes. And then each chromosome is made from very tightly wound DNA molecules. Now, a gene, which is probably the single entity that we discuss most in genetics, really doesn't have a concrete term, or a concrete definition so much as it is just any stretch of DNA, any length of DNA, could be very short, very long, depending on the gene. All it needs to do is code for a particular protein, a particular thing that your body builds. And that is what a gene is. And so we have about 20 to 25,000 genes, which is far fewer than some other animals. I think it's far fewer than like the roundworm. But we have different mechanisms that basically allow us to use our genes for multiple purposes, making multiple proteins out of a single gene. Now, alleles is another important term to remember, and it's probably not a term that most people are familiar with. It's not something you use in day-to-day language. But alleles come from the fact that humans, you and I, share 99.5% of our genome. So we are 99.5% identical. Now that 5%, that 0.5%, half of 1%, is really kind of what makes us who we are. It codes for everything about our physical nature, everything about how we look on the inside, how our organs behave, how our cells behave, how our neurotransmitters behave. All of that is in that 0.5%. So what an allele is an allele is a single base, so a single letter in that instruction manual that varies or is different among members of the population. So if there's a specific spot in our genome that you and I could be different on, if it's something that not all humans share, that's an allele. And so an allele usually means you can have several different variations of it, and that's what makes people different. So we'll talk a lot about different alleles predisposing people to different traits or different mechanisms. Now, some alleles have very strong effects. These people tend to be at very high risk for developing an associated disease. So a couple you may have heard of include things like BRCA1 for breast cancer. If you have breast cancer in your family, you've probably heard that term. That's a gene that puts people at very high risk of developing breast cancer. There's also ApoE4. That's an allele for late onset Alzheimer's. If you have Alzheimer's in your family, you may know of people who are being tested for this allele. So the allele is a gene that varies in the population, and you can usually have a couple different versions of this allele. Now, those alleles are very strongly affected in terms of behavior, but most alleles actually have very weak effects. And most of the traits that we think about in psychology are really complex and have many genes influencing the traits. So we don't really have a gene for neuroticism. You know, maybe there's 100 genes that impact just a little bit, put you at a little higher risk for neuroticism. So we don't talk a lot about these high risk genes in biopsychology, but in the genetics class, you certainly would. Now, there are some traits that are known as dominant recessive. Now, if you take biology and you've done a Punnett square in your life, this is what we're talking about here, dominant recessive. Sometimes these are called Mendelian, and that's after Gregor Mendel, who was the monk who originally studied the inheritance of traits in pea plants. And so that's where the term comes from. But these are really common in sort of rare medical conditions. And these dominant recessive alleles can be split into dominant and recessive alleles. The dominant usually are the more influential of the two, meaning they sort of exert their effect if they're present. And so in this case, you only need one to completely control the trait. So obviously you get 23 pairs of chromosomes. So you have 2 of every gene in your body. One you got from your mom, one you got from your dad. So if you have a dominant trait and you get that dominant gene, you only need to get it from one parent in order to have the phenotype. Now, recessive alleles, on the other hand, or if a trait is linked to a recessive allele, it means you have to have 2 copies of that recessive allele in order to inherit the trait. If you only have one, you don't have the trait at all, and you would be called a carrier. So a lot of people talk about maybe genetic diseases, and if they're a carrier for them, it means they have the allele and they could pass it on to their children. But they don't necessarily have the trait because they don't have 2 of those alleles. Those are what are known as recessive. So a way to think about this is in this graph, and here we have eye color. We have brown eyes and blue eyes. Now this, forewarning, this is a little more nuanced. It's actually quite a bit more nuanced, eye color is, than just blue versus brown. But for the sake of this diagram, we're going to say that there are 2 different alleles that you can have in this gene. So there's we're going to label the first allele with a big B, and that is the dominant gene for brown eyes. You can have a little b allele. We're going to call that the blue-eyed allele for blue eyes. So you can see that the mom and the dad both have one big B, the dominant allele, and one little b, the recessive allele. Now, because the big B is dominant and it's for brown eyes, they only need one of that brown-eyed dominant allele in order to have brown eyes. But because they have these little b's, they're technically carriers for the blue-eyed allele or gene. And so what this means is that if they have 4 children, on average, they're going to have 3 that also have brown eyes and 1 that has blue eyes. And that's because you can see the various combinations that these 2 parents can make. The first child has inherited a big B from dad and a big B from mom. The second child has inherited a big B from dad and a little b from mom. The third child has inherited, say, a big B from mom and a little b from dad. And the fourth child is the only one that has inherited a little b from mom and a little b from dad. And so only in that case, about 25% of cases where both parents are a carrier, will the child have that recessive phenotype or that recessive trait. So some recessive traits are things like cystic fibrosis, sickle cell anemia, Tay-Sachs disease. These all tend to be more common in one specific population. And the reason that they're more common in a specific population is because it has to do with ancestry. So there was an ancestor in this population, sort of a common ancestor way back, and a mutation happened that caused this disease. And that got passed down through the generations in this specific population, usually having to do with a specific group of people that lived in a region. There's also some common medical dominant traits. So dwarfism is probably one of the most common we think of. Dwarfism or achondroplasia is a dominant trait. And so if you have one of the gene, you will be of short stature. But if you've seen any of like the TLC shows, I don't know what their names are, but you'll see that a lot of dwarf parents have full regular height children. That's because the regular height gene is actually the recessive allele. And so two dwarf parents may have a full height child. Now, you may have heard me use these words already, and if you didn't know what they were, I apologize. I tend to use them as if everyone knows them, but they're genotype versus phenotype. And this will be talked about in this class for sure. They're big words for really simple concepts. Genotype is just in the person's entire genetic code. It's called their genotype. It may even be called their genome. It basically just means the sum of all of your genetic information. Now, phenotype, on the other hand, is more about the observable characteristics of a person. This can be anything. It can be your appearance, your hair color, your eye color, your skin color. It could be more complicated things like your personality, your intelligence, aggressive behavior, anything like that could be a phenotype. And so we'll talk a lot about genotypes that predispose you to have a certain phenotype. So they're sort of two opposite ends of the spectrum. Now, heritability, which is a mainstay of my research, heritability is something people talk about a lot. I'll say that almost every trait you can imagine is heritable. Even things as complex as, religious service attendance are heritable. Heritability is simply a statistical measure. And what it is, and this is the complicated way of saying it, it's saying the proportion of phenotypic variance. So it's the differences between people in how they appear or act or behave, those differences, and how much those differences are due to genotypic variance. So the differences between their genes. So how much of a phenotype in the population is due to genotype in the population? So you'll see estimates usually between 0 and 100% for heritability. Some psychiatric traits are highly heritable. Things like schizophrenia, bipolar, autism are all highly heritable on the order of 80-ish%, which is just about as high as diabetes and heart disease. So it's right up there with the same heritability as many medical conditions. Then other traits are more equal heritable versus environmental influence. Things like depression. Depression is usually estimated around 40 to 50% heritable. And that just gives you sort of a general estimate of how genetic a trait is, how influenced by our biology that trait is. The higher the number, the more influenced by biology. Now, the way that we calculate heritability is through twin studies. And in twin studies, we use two different types of twins. So the first type of twin is called an identical or a monozygotic twin. So the reason it's called monozygotic is the word zygote, Z-Y-G-O-T-E. Zygote is the name for a fertilized egg. Mono means one, so from one zygote. And you'll see this in the description here. So a monozygotic pair of twins occurs when a single fertilized egg, or zygote, divides into two separate entities. So you basically got one egg and one sperm. They fertilize, and then once they've formed basically a zygote, the very first stage of a human being, they split. So they are identical. They are from the same DNA. So they are genetically identical. Monozygotic twins, with the exception of some rare mutations that happen after that division, share 100% of their genes. And because of this, they will always be the same sex because they share those same sex chromosomes. Now, dizygotic, on the other hand, are what is known as fraternal twins. And dizygotic twins, the word di comes from two, and zygotic comes from zygote. So it actually is two separate fertilized eggs. So in this case, the female releases 2 eggs, or ova, instead of the normal one that's released each month. Those 2 eggs are fertilized by 2 separate sperm, and they form into 2 separate individuals. So basically, these are just siblings that are born at the same time. They're no more similar than you and a sibling might be. Just happen to be born at the same time. Now, because they're just as similar as any siblings, they can be the same sex or they can be opposite sex, just like 2 siblings can be. So when we think about heritability, and this is just something from sort of what I do in life, is usually we see sort of a model like this, where you have a trait and you have these 3 parameters that can influence the disease. So you have genetics, you have we call this an ACE model, which basically just means we split the trait into 3 parts that can contribute to it. The first is genetics, and that's A because it stands for additive genetics, but basically the A is heritability. There's also 2 different types of environment we can think about. So we can think about a shared or a family environment, things that you and your siblings share, you know, what you eat for dinner, the parenting style of your parents, the neighborhood you live on, and then also unique environments. So things about you that are different from your siblings. So maybe the sports you play, or the teachers you have in school, or your peer group. That would be unique environment, things that are unique to just one person. So what you do in a twin model is you get correlations. And if you've taken statistics, you'll know that R is the letter that denotes a Pearson's correlation. So here what we have is 2 formulas. We have RMZ for monozygotic and RDZ for dizygotic. So you have the correlation for monozygotic twins and the correlation for dizygotic twins. So how similar? Say you're measuring height. Let's do something really simple. Actually, height is incredibly complex in genetics, but let's say you're measuring height. How similar are monozygotic twins? Is it a really high correlation? Probably. What about dizygotic twins? What's the correlation for them? And you get those numbers, just like you would do a correlation for any other self-report measure that you've done in stats or in research methods. But what's cool about twins is that we know what contributes to those correlations, because we know exactly how much genetics they share. We know exactly how much shared environment they share. We know exactly how much unique environment they share. We know how much A they share, genes, because MZs share 100%. So we just have A in their formula. And we have half of A for DZs because they share half of their genes. We know that both MZs and DZs share their shared environment. It's called the shared environment because they share it. So we have a full C in each equation. And we know that both twins don't have any overlapping unique environment because by definition, it's the stuff that's left over. So we know that the MZ correlation equals A plus C. We know that the DZ correlation equals 0.5 plus C. And this is not a formula you need to know, but this is just so you understand what heritability means. And so we can basically take those 2 formulas and calculate the value of A, C, and E, and we can pull out of that very easily. I mean, if you give me a data set with twin data, I can get you the heritability of a trait in probably 10 minutes. If I have to, you know, remember how to get back into the statistical program. It's very simple. You can get the calculations for A, C, and E. So you can determine how much of the trait is due to genes, how much is due to common environment, the family environment, and how much is due to the unique environment. And that's really a lot of what I do in my office when I'm not teaching. So now you know. So another basis of genetics that is important for psychology is evolution, and specifically evolutionary theory or evolutionary psychology. Evolutionary theory is sort of a broad scientific principle. It's used in psychology, it's used in biology, it's used in all sciences. And this is obviously made famous by Charles Darwin. This is an English biologist. He wrote On the Origin of Species. He was also pretty famous in the field of developmental psychology, too. If you take a developmental psychology class with me, you'll hear me talk about him writing a whole book about his, newborn son and the developmental milestones he hit. So he wasn't just a biologist. But The Origin of Species was really the main book for evolutionary theory. And the main thing it sought to do was determine how species change. And Darwin said that nature really works to ensure that species do two things, two very simple things: survive and reproduce. And that's really the basis of evolution. It's actually, people think of evolution as this complicated theory and system, but it is so simple. And I'm going to try to make it as simple as possible for you. So when we think about evolutionary fitness, what we're talking about is really the fitness that is discussed in the principle of the survival of the fittest, which is often a principle you hear related to evolution, but it's honestly one of the most confused principles out there. Survival of the fittest is really the single basis of Darwin's evolutionary theory. It's also a very simple concept, but one people often misunderstand. So what evolutionary fitness has to do with is how well you can pass your genes on to the next generation. In other words, how many children you have. That's really what drives evolution. So what the theory says is that traits that increase the amount of children you have will also be passed along to the next generation because they share your genes. So if you have a trait that makes you super fertile and you have 30 children in your life, that trait is going to be more represented in the next generation because you had 30 children. You can think about it in an opposite way. So maybe you have a trait that makes it very hard for you to have children. It can be a behavioral trait. Maybe you have a psychological illness where you have less success in mating. Maybe you have a physical ailment that makes you have less success in mating, and you have less children. Now that gene will be less likely to appear in the next generation because you had less children. So it's not just obviously one person, but on a population scale, a trait that increases fitness will be seen in successive generations because there's more children produced with that gene. It's really a simple concept. And when you start to understand evolution in that basic of a principle, it kind of, everything falls into place and makes sense. There's actually a book called The Selfish Gene that discusses this. It's, it's a little old at this time, but the principles are the same. It really talks about how the gene is selfish. It's all about reproducing and surviving. So like I said, this is the entire basis for what some people call evolutionary selection. So a trait is either selected for or against. Traits are selected for if they increase the fitness in the population. Fitness meaning the ability to reproduce. They're selected against if they increase or they decrease fitness. So a person with that has low fitness or produces less children. Here's an example of what I was talking about, where you've got an animal with low fitness and an animal with high fitness. And if you have twice as many children in, a couple generations, you're having an exponentially more amount of that trait in the population and that gene in the population. So genes that make you reproduce more also get passed down more and become more prevalent in the population. Evolutionary psychology is a branch of psychology that uses evolution to theorize about why certain traits are common in humans. So some of these traits may not be intuitive. So one trait that is has been researched a lot and is kind of an interesting paradox is sickle cell anemia. So sickle cell anemia is a genetic trait that is common in African ancestry, and the red blood cell is not shaped like a circle, it's shaped like a sickle. It's sort of like a half moon shaped. And this design of the blood cell actually causes a lot of symptoms because they can clot easier and will cause a lot of cardiovascular issues. But it's pretty prevalent in the population. And so evolutionary psychologists or theorists would think, why did a trait like this stick around? Wouldn't that have decreased the amount of children someone would have if they were dying before childbearing age? But what's interesting about a gene like this is that there's been some research showing that the same trait, sickle cell anemia, and remember, it's common in people of African ancestry, so that's where the gene mutation happened, was probably in the population living in Africa, was that this also actually protects somewhat against malaria. And so it may have had a detrimental effect to some people, but it also may have helped people live longer as well. So that's just one example to say how, traits are complicated. And usually if a trait sticks around in the population, there's some advantage that it's giving in terms of fitness, the amount of children people are having. And if you're interested in evolutionary psychology, Dr. Fay teaches a class, I think every semester, maybe even 2 classes, evolutionary psychology. And I'm hoping to be teaching behavior genetics, more about the molecular side, in the next year or 2. So there's definitely opportunities to extend this class if you love biopsychology. So here's another sort of, what are we most afraid of? Sort of some things that can be counterintuitive. If I were to ask you, what are you most afraid of? Most people would say the snake, not the car. Now, is that an accurate fear? Should you be more scared of the snake than the car? No. Cars kill by far more people than snakes. But not a lot of people have this sort of innate fear of cars. But a lot of our traits come from what was helpful, not necessarily to us, but what was helpful to our ancestors, what created our ancestors' ability to be more fit, to have more children, and that was passed down to us. And so maybe there was a benefit of having a fear of slithering objects, things that moved in an odd direction, like, the odd patterns that snakes make. Maybe there was an evolutionary benefit of being scared of that movement. Maybe it meant you got bit less from snakes, and those people that had that gene had more children just because they stayed alive. Does that mean that gene got passed down to us? Maybe. That might be why a lot of people are scared of snakes and spiders, but we don't have that innate fear of cars. And that's the kind of thing that is hypothesized in evolutionary psychology. So the final topic I want to talk about is complex genetics. Now, I use the term complex genetics. Some people say complex trait genetics, but basically, most traits are complex. Now, some of the diseases we talked about have those single gene disorders, dominant recessive, but most traits are not like that. So most traits are polygenic, which means they're affected by a lot of genes. They're also additive, meaning the genes with very small effects add up. So you might have 100 genes, all with very small additive effects. And they're also, the traits tend to be multifactorial. And what that means is not just influenced by genetics. So they might be influenced by genetics, your common environment, your unique environment, the prenatal environment, interactions between all of them. Basically, it's complex, it's complicated. And really the one way that we have had the most research in thinking about this complex genetics in the last 10 to 15 years has been the field of epigenetics. And epigenetics is probably something you will hear about. It's kind of starting to pervade our psyche in terms of how we think about the environment. So epigenetics is basically the way that our environment influences how our genes are expressed. And to some that sounds counterintuitive because you're born with your genes, they don't change. And you're right, they don't change. But what changes is how your body expresses those products. Remember, DNA is like an instruction manual telling your body how to make things. And epigenetics, or these environmental influences, can change whether or not your body sort of really actually makes those things that your instructions are telling it to. That's called expression. So your body can say, hey, make 50 of these proteins, but some other things can affect whether or not that process goes well and whether you actually make 50 of those proteins. So one example that you see a lot of research on is the field of stress, because let's admit, we all undergo a lot of stress. So stress is an environmental experience, and it can add a chemical marker on your gene. So it's basically, if you've heard of oxidative stress, it's a chemical process happening with stress that changes the chemical makeup of certain cells. And so little chemical markers called methyl groups can be attached to our DNA. And this essentially makes the instruction manual harder to read. So it's kind of like if you had a book, your instruction manual, and you glued 2 pages together, the methyl group glued 2 pages together. You wouldn't be able to get in there and read the instructions, and so you wouldn't make that protein. Sometimes you'll hear them as like genes turned on or off. Basically, this is an example of what unmethylated DNA would look like. So the blue is DNA. It's wound around these little proteins called histones, and it's kind of loosely wrapped around these proteins. That's what normal DNA looks like. But when it's methylated, it scrunches up all tight together. And you can imagine if you were trying to get in there and read all the base pairs on those blue lines, it would be a lot easier to read that blue line if you were looking at this unmethylated DNA versus trying to get in there and pull apart all these little proteins to get to the DNA in the methylated DNA. So you might have the gene to produce this certain neurotransmitter, say serotonin, in your brain. But if you have this methylated DNA, this experience has made it harder for your body to actually produce that. So genetics is complicated. And there's a lot of really interesting research going on. In fact, genetics has only been around for, say, 50 years, that we've even known what the structure of DNA is. And so much has happened in 50 years. So these are all the processes that are really underlying all the stuff we're going to talk about in the book. And we won't go into a lot of detail about these topics moving forward, but it's good to know sort of what is underlying all the brain chemistry we talk about, all the brain anatomy that we talk about, all the hormones we talk about, all come from these processes. So if you have any questions for me, there's a couple ways that you can ask them. This is our first module of Real Materials. So the first way you can do, and I actually recommend this way, is to go to the discussion board. There's a discussion forum that just says ask a question. This is a great place to ask a question because if your classmates have the same question, they can find the answer there. And so if you have a question, you might also want to go there, see if anyone else has asked it, and if they haven't, asked it, and I will come and answer the question and the whole class can see it. If you'd rather email me, you can. I mean, if it's a very specific question just about something important to you, you can email me. I also have office hours on Thursdays through Zoom. You can find the link on the left-hand side of Blackboard. So every Thursday, I'll be in a Zoom room. If you want to come in and chat and ask any questions, you can. So any of these 3 ways are good ways to get in contact with me if you have any questions. And I hope to hear from some of you. If not, I will see you in our next modules.